Hello everyone, we will have a revision on carbohydrate metabolism, glycolysis. So the question will be like this: outline the reactions of glycolysis, calculate the energy yield during aerobic and anaerobic glycolysis, which usually comes as an essay. Even they can ask this question as for short note also. On that time they will be asking only outline the reactions of glycolysis. Okay, let's see in detail. So in your answer, it should have a specific definition. So you should define that what is this glycolysis. We know that glycolysis is a oxidation of glucose molecule into pyruvate aerobically, anaerobically, which will be converted into lactate. That is the definition. Then where does it occur? That means the site, the organs and subcellular organs. So site means almost all the cells, in almost all the cells that glycolysis occurs, and subcellular site is cytoplasm. And you can just mention the key enzymes of glycolysis like hexokinase, phosphofructokinase and pyruvate kinase. And sometimes even you can write the significance of glycolysis. Why does the body needs this glycolysis, or importance of glycolysis also can be mentioned. Then straight away you can draw the pathway of glycolysis. Once you draw the pathway perfectly with enzymes, coenzymes and substrate, intermediates and the final product, etc., no need to write in detail about the step-by-step reaction. Further, depends on the question. In this question, they have asked about the energetics. That means the how much energy is produced during aerobic and anaerobic glycolysis. But sometimes they can even ask the regulatory part also. If asked or not asked, you can have a mention regarding the regulation. That is up to you. So this slide which explains the whole pathway, we know that the 6-carbon glucose molecule will be finally oxidized into 2 molecule of pyruvate, or depends on the situation, which is even lactate. So glucose undergoing a phosphorylation reaction with the help of hexokinase, where ATP hydrolyzed into ADP to form glucose 6-phosphate. Further, an isomerization reaction occurs to form fructose 6-phosphate with the help of phosphohexose isomerase. Then one more phosphorylation occurs with the help of another enzyme, phosphofructokinase, where ATP will be giving its phosphate group and itself will be converted into ADP, and the product is fructose 1,6-bisphosphate. For both hexokinase and phosphofructokinase, the coenzyme is magnesium ion. Now this up to this fructose 1,6-bisphosphate, it is a 6-carbon molecule, 6-carbon monosaccharide. Now this will be breakdown into 2 molecule of 3-carbon subunit as dihydroxyacetone phosphate and glyceraldehyde 3-phosphate, and the reaction catalyzed by aldolase enzyme. This reaction, this dihydroxyacetone phosphate can be converted into glyceraldehyde 3-phosphate with the help of phosphotriose isomerase enzyme, which is a reversible reaction. So hereafter the molecule will be, the further reactions will be two molecule of glyceraldehyde 3-phosphate will be oxidized to 1,3-bisphosphoglycerate with the help of glyceraldehyde 3-phosphate dehydrogenase, where NAD acts as a coenzyme. NAD will be converted into NADH. Usually this NADH can enter into ETC and can produce 3 molecule of ATP. This occurs during aerobic glycolysis. Now this 1,3-bisphosphoglycerate undergoing a dephosphorylation reaction from 1,3-bisphosphoglycerate, 1-phosphate will be given to ADP and ADP will be converted into ATP. So this is done with the enzyme phosphoglycerate kinase enzyme. So remember, this is a substrate level phosphorylation. Now this 3-phosphoglycerate undergoing a mutase reaction with the help of phosphoglycerate mutase to form 2-phosphoglycerate. Now there is only a single rearrangement of phosphate group occurs in this reaction. The phosphate group on the 3rd position will be shifted to the 2nd position. Now the 2-phosphoglycerate will be converted into phosphoenol pyruvate with the elimination of a water molecule, and the enzyme here it is enolase enzyme, and this enolase enzyme which requires magnesium as the coenzyme. And the importance of this step is that Enolase, the metal fluoride can inhibit enolase. So usually while collecting blood sample for the blood glucose estimation, we used to take the blood sample in a fluoride bulb or fluoride bottle. Why to? Why? Because this fluoride can inhibit enolase, so that which can prevent the further glycolysis after the blood collection. Or else what will happen after, means it will take some time to for the blood glucose estimation. By the time what will happen, the live cells will utilize the glucose and we will get a low value uh, during the time of estimation. Okay, that is the importance of this step. Now this phosphoenol pyruvate will be converted into pyruvate by another substrate level phosphorylation. From phosphoenol pyruvate, the phosphate group will be removed to ADP and ADP will be converted into ATP. So these two are the substrate level phosphorylation, phosphoglycerate kinase and pyruvate kinase reaction. So up to this, we have got out the final product of glucose oxidation as pyruvate. So this much occurs during aerobic glycolysis. During anaerobic glycolysis, what will happen? The pyruvate will be reduced to lactate with the help of lactate dehydrogenase enzyme. So now for this reduction, we need NADH. This NADH will be utilized with the same step as glyceraldehyde 3-phosphate dehydrogenase. So what will happen in during anaerobic glycolysis? This step does not occur. Okay, so this is all about glycolysis. Now this is regarding the energetics. So in question we have seen that we have to explain the energy production during aerobic condition and anaerobic condition. Aerobic means whenever the oxygen is available. So we have seen first and third reaction, that is your hexokinase and phosphofructokinase reaction, where ATP was utilized. So we have to minus 2 ATP molecule from the overall reaction. Now the glyceraldehyde 3-phosphate dehydrogenase, which is the oxidative phosphorylation, occurs here. Here where the NADH will be entering into ETC and one NADH can give 2.5 ATP. Okay, so here 2 molecule of glyceraldehyde is going and converted into finally pyruvate. So we have to multiply the 2.5 into 2. You are getting 5 ATP. And another two reaction, that is 1,3-bisphosphoglycerate kinase and pyruvate kinase. These two are the substrate level phosphorylation, where ADP was converted into ATP and the phosphate group was yield from the intermediate. So we are getting 2 molecule of these intermediates will be converted and 2 molecule ATP each. So 2, 4 plus 5, 9 ATP total, and we have to subtract the first 2 which we have utilized in the initial reaction. So 9 minus 2, total 7 ATP is generated during aerobic glycolysis. Now this is regarding the anaerobic glycolysis. Anaerobic glycolysis, what will happen? Already we have discussed that the pyruvate will be reduced to lactate, and we have seen that the NADH which synthesized in the 2,3-bisphosphoglycerate, that 2,3-bisphosphoglycerate dehydrogenase will be utilized here for the reduction of pyruvate to lactate. So what will happen? 2.5 to 2, that is 5 ATP will not be synthesized here. Remaining same, the hexokinase and phosphofructokinase utilize 2 ATP, so minus 2. Then 1,3-bisphosphoglycerate and pyruvate kinase, that is our substrate level phosphorylation, 2 plus 2, 4 ATP is generated. 4 minus 2, only 2 ATP is overall synthesized during anaerobic glycolysis. So this figure which explains the regulation of glycolysis. So the major hormone which regulate glycolysis or which enhance glycolysis is insulin. So insulin, which helps to stimulate the 3 glycolytic enzymes as hexokinase or glucokinase, phosphofructokinase and pyruvate kinase. Insulin furthers the reaction and glucagon inhibit the glycolytic enzyme. Insulin stimulate the glycolytic enzymes. So this is which explaining how individually each enzyme how to help for the glycolytic regulation. These are the activators and inhibitors. Hexokinase enzyme, which is usually inhibited by glucose 6-phosphate itself. Then glucokinase enzyme activated by insulin, inhibited by glucagon. phosphofructokinase, which is the rate limiting step of the glycolysis or rate limiting enzyme, which will be activated by several factors like insulin, AMP, fructose 6-phosphate, phosphofructokinase 2, another enzyme, and fructose 2,6-bisphosphate, etc., which will be inhibited by glucagon, ATP, citrate, low pH, cyclic AMP, etc. Okay, so phosphofructokinase, the enzyme will be regulated by allosteric modification. Then next is pyruvate kinase. PK is for pyruvate kinase, which is activated by insulin and fructose 1,6-bisphosphate, inhibited by glucagon, ATP, and cyclic AMP. And the last one, PDH, that is pyruvate dehydrogenase complex, activated by coenzyme A and NAD, inhibited by acetyl coenzyme A and NADH. Thank you. That's it. This is all about glycolysis, how to write an essay. Please study thoroughly.